So where we left off on was antibodies. This is the slide that I finished up on Tuesday, was looking at the different things that antibodies can actually do. We typically think about them as doing the opsonization, which is what we started at with chapter 14, right? Which is that antibodies basically bind. Notice now that we learned a little bit in chapter 15 about the antibodies, where the two legs essentially are the antigen binding regions, right? So that's the variable part. Those vary, so that way they can bind to all the different types of antigens that we potentially can find inside the body. And then the constant portion, the actual stalk, is got to stay the same on all antibodies and to ensure at least all the classes. So there's 5 different classes of antibodies. The classes have different constant regions, but for the most part, the constant regions are the same. And the reason for that is that way things like the phagocyte here can actually recognize and bind. That part has to stay the same so that the phagocyte's doesn't have any business knowing all the different antigens. Its job is just to come and engulf and destroy, whether it be the macrophage or the dendritic cell. That's its job. So we've talked about this in chapter 14. Chapter 15, the complement system, if you go back and you actually looked at some of the videos on it, I didn't get into all of it because it does get complicated, but you may have seen that there are those different complement proteins start as antibody-based. So there is that C1q, whatever the heck it is, antibody that actually starts the whole complement system cascade. Is an antibody that binds up and starts that process. But again, I'm not asking you to be responsible for that part of the complement system. Here, these two are more or less the same, as well as neutralization, but they all have their own little meaning. So immobilization, if you're immobilized, right, you can't move. So in this case, these are going to be for bacterium that have flagella that are able to move around. And then so the antibodies, remember, flagella is one of those pathogen-associated molecular patterns. Those are the ones that we recognize over evolution are bad. And so in this case, these antibodies are immediately going to start picking up the flagella. And they're going to start cross-linking. Having two hands, I grab one with one hand, grab one with the other, now they're stuck, right? So that's what the antibodies are doing. They're grabbing different parts of the different bacteria, basically kind of just making a big mess. Yeah.
So, is that the name of the antibody that does that?
Which one?
The
immobilization, that would be like a class of antibodies that do that.
Okay, so it's a type.
Yeah, it's just a type. Then cross-linking, notice the difference between them is cross-linking, they don't have flagella. So at this point, we're just blocking them up because a lot of the bacteria, while they do grow in colonies, remember they multiply by binary fission. So they're basically making clones of themselves. So they pretty much stay in the same area, especially if they're not mobile. In order to actually cause disease, sometimes they actually act individual to be able to get across the epithelial layer, get across maybe the membranes of the cerebrospinal fluid, whatever it might be, to get over to the other side. By having them all cross-linked together, they can't go anywhere, right? There's one big mess. Then the third one, neutralization. Notice that's for extracellular viruses and toxins. And all we're doing is blocking the toxin and virus's ability to actually cause problems by basically binding all around it, essentially, blocking it. The third one, or the fourth one down here, is antibody-dependent cellular cytotoxicity. So a big mouthful, but the name tells you what it does. It's antibody-dependent, so it requires antibodies. Cellular cytotoxicity. Whenever you see the word cellular cytotoxicity, think about the NK cells, think about the CD8 T cells, because their job is cytotoxic, right? So the NK cells, their job is to come in and kill any infected self cells. So any of our cells that are infected with virus, or there are some bacteria that get inside the cells, their job is to go recognize that by the antibodies that bind, and recognize again. Notice what it's actually binding to. It's binding to the FC region, right? It's binding to that constant region of the antibody because it's able to recognize it. Recognizes that the antibodies bond there. It sends a signal to the cell to induce apoptosis. Remember, the reason that we do that, twofold. One, we want to So one reason we do apoptosis is to get rid of that cell, right? Because if we kill the cell, any of the viruses that are in there are going to die too, or they're not going to be able to be, capable of causing disease. Second thing is, it's just better for the greater good to knock out that one infected cell than it is to allow it to live, and feel bad for it, right? Oh, we don't want to kill our own cells. Well, yeah, we do. We want to get rid of them, especially if they're infected. And by going through apoptosis, it keeps the inflammation down. That's the whole key for apoptosis. If we had the cells just dying, if that virus actually did grow up really large numbers and then burst through the cell, that would cause the cell to basically die uncontrolled. That would cause inflammation to increase. So by having NK cells come in, induce that apoptosis, it allows the cell to basically go through an organized cell death that minimizes inflammation. So here's a slide that just goes through all those exact things I just talked about on the cartoon. Just to help you, remind you if you don't recognize it right away from the cartoon and the notes you're taking. All right, so now we're going through the 5 different classes of antibodies. There is the IgMs, IgG, IgA, IgD, and IgE. Each of them have their own role to play in the immune system. Remember, antibodies' old name was immunoglobulin, so that's where the IG comes from. It's just an inherited name, essentially, that's been passed down and hasn't changed. They all have the basic monomeric structure. So when we're saying monomeric, that means what does one antibody look like? Well, they all look like what we've already been discussing, right? They all have the 2 legs and the 1 stalk. Each class has a different constant region. So here, the constant region, right? The FC region, that's the constant. The 5 classes are going to have a different constant region because each class has its own role to play. So it's going to be having different parts of the immune system respond to it. So the constant region is going to be different amongst the 5 classes. Some form multimers, some don't, and we'll see that as we go forward. So multimer is when the antibodies come together to make a bigger structure. And so each of the different classes have a distinct function and property, as I've already said, it says there too. So this is a good slide to star or whatever for go back and look. It tells you everything you need to know in terms about the 5 different classes, and then we got 5 slides after it that's going to go over each of the classes on its own. But what we have here is we got the IgM. Notice what it's doing there. It's making a pentamer. So it's actually got 5 IgMs all together. And it just basically rolls around looking for anything. So it's maximizing the number of spots that it can bind to what it's looking for. And so this is the first antibody class produced during the primary response. Remember, primary is the first time you get sick with that particular disease. So IgM is the first antibody class produced, principal class produced in response to T-independent antigens. So remember, these are the ones that don't need any help. That's going to be the same thing as the pathogen-associated molecular pattern. So it's going to be the peptidoglycan, the LPS, the flagella, the double-stranded RNA. Doesn't need any help because those are ones we recognize over time through millennia as being bad. Provides direct protection by neutralizing viruses and toxins. Binding of IgM to antigen leads to activation of the complement system. So IgM has lots of roles to play, but it's in that immediate primary response. IgMs become useless once we start progressing the adaptive immune response, but they're the ones that are there from the get-go. IgG is the majority of your antibodies. So when we're talking about antibodies, we're usually talking about the IgGs. And so you can see that here, percent of total serum. So when we take somebody's serum and we look at the white blood cells and we look at the amount of antibody being produced, look at the percentages there. 80 to 85% of all the antibody that you have in your serum is IgG. So it just tells you right there that that's probably the most important. IgA, notice it makes a dimer. So there's 2 of them connected together. These are important for your mucosa protection. So anything that's around the mucus, this is going to be the most abundant class that's produced. And so this is important for inducing any sort of immunity to food, like getting some sort of food poisoning type infection, something that might be up in the nasal cavities or down through the esophagus, or even into the lungs. The IgA plays a big response in terms of mucosal immunity. IgD and IgE. IgD, notice how small a percentage it is, less than 1%. We're not 100% sure what it actually does. We know it exists. We've been able to identify that it is a separate class of antibody, but truly understanding what its role is and why it's important, even at those very small percentage amounts, is pretty, it's still out there for somebody else to figure out. And in IgE, these are ones that are responsible for your allergic reactions, as well as detecting parasites. And when we use the word parasite, we're typically talking about something that is macroscopic versus micro, right? Micro is something we need a microscope to see. Macro, we can see it with our own eyes. So when we use the word parasite, we're talking about something we can see with our own eyes, so like worms. So that gets a worm infection, which is very nasty, but when it does happen, IgEs are responsible for that. So any sort of extracellular antigen, such as an allergen or a parasite of some sort, the IgEs are going to be responsible for that. Yep.
What is the significance of the days next to it, the half-life in the serum? What is that?
Tells you how long they actually survive, like how long are they actually around. So that's also going to get your bloodstream. Right. Remember, we can't keep all these antibodies, we can't keep all these immune system cells around indefinitely. They go down to being just a few that get kept behind as memory. But once the antibody response gets rolling, how long do we actually see it in the bloodstream? And that's what we're seeing there in those numbers. Good question. All right, so then, the next 5 slides are just going through each of the individual antibodies on their own. So a lot of what I just said is already still applies in these slides. IgM, roughly about 10% of your circulating antibodies. Remember, it is the first ones on the scene. It's the first ones that are just floating around in the bloodstream looking for those T-independent antigens, the ones where we don't need any extra help. They make a pentamer, so it gives it 10 antigen binding sites, which is good. It aggregates very effectively. So when we go back to aggregation, that is the idea of this part right here, where we're basically binding them all up. That's aggregation. So we're aggregating, we're basically binding them and locking them all up. So having 10 antigen binding sites is even better, because I can bind even more, and I just start, basically putting them all, basically getting them all as immobilized as possible. Large size prevents crossing from bloodstream to tissue, so this is problematic. We only find the IgMs in the bloodstream. If we need to get antibodies over to a site of infection that's on the other side of the bloodstream, then we need the IgGs, the IgAs, or the IgEs to be involved. It's the most efficient class at triggering classical pathway of complement. So remember, there's 3 parts. There's 3 ways that complement can be activated: classical, alternative, and mannose binding. And so the IgMs are the ones that help activate the classical pathway of complement. All right, IgG though, make sure you star this one. IgG is your main antibodies when we're talking about antibodies in general, and the ones that are being produced by the plasma cells, right? The B cells mature into plasma cells. Plasma cells are the ones that are actually producing antibody. The ones that they're producing the most of when we're talking about an individual infection is the IgGs. So they're going to be 80-85% of your total serum immunoglobulin levels. They can exit the blood vessels, go into the blood, go into the tissue. They can get anywhere they need to be because they are just a monomer. They don't bind up in the dimer or pentamer shape. Produces the longest term protection. Half-life is 21 days. So as we move forward, we'll talk about how your antibodies in your system will stick around for a few months. They slowly are weaning away, but they do stick around for a little while in case that infection comes back immediately, which is also good for maternal to child. So when a child is born, the mom's antibodies are still in the child for upwards of about 6 months while their immune system is kicking on. And so this is important too, that they have that long half-life in order to survive for a decent period of time while the child's immune system is coming online. As I already said, first and most important circulating class during the secondary response. So basically, Once the infection is seen again, IgGs just come in and tear it apart. It's pretty quick for them to come back online from the memory cells and immediately start taking out whatever it might be. It can protect through pretty much every single way that we've seen there in that first cartoon that we started with today. That's all that fourth one is pointing out each of the different classes of protection that antibodies can do. And as I just pointed out, transported from placenta to the fetus bloodstream, right? So we can actually move it back and forth. This is something we're going to talk about a lot as we move forward too, is that the placenta acts as a very good barrier because that child is foreign to mom's immune system. And so the placenta basically acts as a good barrier to protect mom's immune system from taking out the child as it's developing. So very little can actually get across the placenta, but the antibodies can. Unfortunately, some pathogens can, and we'll talk about that more as we move forward. But these antibodies can. They give the child some protection against pathogens that the mom's already seen. So maternal IgG protects fetus and newborn, as I already said, degrades gradually over a 6-month period if it begins producing their own. So this graph here shows you that happening. So right here is birth, right, about 9 months. And as we move over here, the purple is the total IgG that's found in that child. Well, you can see it ramping up all the way up to birth. Well, when we start separating out mom versus infants, what we start to see is right at birth, the child's IgGs start coming online. Not in high amounts because they're just starting. Moms are there in high amounts still at that point. As moms start weaning off with that 21-day half-life, as mom starts weaning off, child is starting to come on, and hopefully that's enough to protect the child. Right? So a lot of what we talk about as we move forward is going to be that initial reaction. A lot of times, the fight is won in the very beginning. So if a child gets sick and they don't have the right IgGs up, we see a lot of childhood death from preventable diseases within the first few months of life, mainly because there's this conflict with the IgG amounts. And so it makes it difficult for the immune system to fight off the infection. And so we'll see those as we move forward into all the different types of diseases. IgAs. All right, so monomeric form is in 10 to 13%, so back to about the same as the IgMs. It's a dimer, so that means, as you've seen before, it has two of them that connect together. And it is the most abundant in terms of the different styles, but for the most part, the biggest thing for you to recognize is what I got bolded here, is that it's the important part of the mucosal immunity. So it's going to be right there at the beginning. The mucosa, remember the mucosa and the skin is the two biggest barriers of the innate system. So IgAs are going to be sitting there in the mucosa. IgMs are circulating around the blood. So they're there and available. So gastrointestinal, general urinary, respiratory tract, anywhere where there's mucus, there will be IgAs. Says here also secretion involves saliva, tears, breast milk. IgAs are found. And protects breastfed infants against intestinal pathogens. So IgAs are important there. And these have only really truly been understood for about 15 years. When I was in microbiology department at Tulane, one of the vaccinologist professors there was working on trying to get a vaccine to induce IgA response. And his argument was, well, what's the point in inducing an IgG response or an IgM response if it's bloodstream when we're dealing with an intestinal bug like E. coli or Salmonella? It'd be better to induce the IgA response since it's found there in the mucosa. And he was right. It actually worked, and it became a successful vaccine for Salmonella typhi. This was something that we're just now recognizing. So again, some of the information, maybe if you go and you start Googling, well, what the heck else can I learn about IgD or IgA? You may not find a whole lot out there because it's relatively still new information. Produced by plasma cells, right? All the antibodies are produced by plasma cells, but this one is telling us produced by plasma cells of mucosa-associated lymphoid tissue. Remember, those are the different neighborhoods that are found throughout the body. So we have the skin-associated mucosa associated were the two biggest ones that we looked at. So the plasma cells that are found in the mucosa are the ones producing IgA, and that just makes sense. You know, that would be the ones that would be most likely to be producing the IgAs. All right, IgD, notice there's only 2 bullets because we don't know nothing about them. There's not a whole lot there that we know. We'll see a couple things that we point out as we go through the rest of the semester that are going to be, well, we know about it. It's important for you to know, just in case we start learning more about it. But at the end of the day, there's not a whole lot we know from the get-go about it. So, involved with development and maturation of the antibody response. Function in blood not clearly defined. So they're there in some sort of fashion to help with the maturation, which we haven't got to yet, but how do the antibody responses mature over time? Why is it the secondary response is faster than the primary? One, we know the memory cells are there, but what we're also going to learn is that there's a maturation process, so in terms of maturing and becoming better. And so what they're saying here is that IgDs are responsible in some way of helping that maturation, but what way is not really defined. IgE, barely detectable in the normal blood. The only time that they're going to be detectable is if you have an allergic reaction or if you are having some sort of parasite infection. And other than that, a normal healthy person will have 0 to 0.01% IgEs running around. But once they get that allergic response, part of that is the histamine, as we talked about before. Also, it's the IgEs being responded or being released to go combat whatever that allergen is. So they're tightly bound to the FC region of the basophils and mast cells. Remember, basophils are important. The eosinophils, basophils are important for the response to an allergen. Antigen binds to 2 adjacent IgE molecules carried by mast cell. Cell releases histamine and other inflammatory mediators. So the IgE is the predecessor to the histamine release. Histamine is what's actually causing you to start sneezing, coughing, watery eyes. That's your inflammatory response being caused by the histamine release. What's causing histamine to be triggered is the IgEs binding to whatever that antigen is. So basophils and mast cells also release chemicals when IgE binds to normally harmless foods, dust, pollens, causing that allergic reaction that, again, is mainly a mistake. You know, there's really no point to somebody having an allergic reaction to something like, well, for me, growing up, I was allergic to green peppers. But why? You know, there's no reason, right? It was just a mistake made by the immune system. But, again, it takes about for me, it took about 10 years of just staying completely away from green peppers. Now I cook with them every day. You know, not every day, but I cook with them quite a bit. Took a little bit of a testing, tip your toe in the water, make sure you're not going to have a problem. But over time, you know, eventually, if you stay away from that allergen, you can usually clear it. Yeah.
So are all allergies like that? Like all allergies, peanut allergies, like grass allergies, those are all just mistakes?
In my mind, they are. I mean, I haven't seen anything out there to tell me why somebody should be allergic to peanuts, why somebody should be allergic to grass or green pepper.
So the immune system just mistakes it for something else.
Makes a mistake for it, yeah. And, you know, it's just the generic randomness of how the antibody response is made. That, for whatever reason, it had the capability of detecting that particular epitope.
And then you said you stayed away from it. Do you think that since your immune system's making a mistake, like the more you're exposed to it, the more immune you become to it?
No, the more you're exposed to it, the more you're going to continue having responses to it. So staying away from that particular allergen can potentially get rid of those memory cells. Again, remember the biggest question is how long do memory cells live? We don't know. Right. So, you know, for me it was about 10 years and then I ended up having 0 reaction to it anymore.
Is that like how like developed reactions work too? Like I just became allergic to seafood like 2 years ago. So is it like the same thing, just like a mistake that just happened?
Yeah. Yeah. And again, it's just because every day you're making new B and T cells in your bone marrow, and every day it's just, we'll see it at the end of this chapter. There's a random what's called VDJ recombination of the genes, and the genes just recombine in all these random different ways. There's no reason for it, and so at that point somewhere along your line, your body created antibodies against seafood by accident. You ate seafood, and they found each other, and you end up having an allergic reaction. That's the easiest way to put it. I mean, it's much more complicated than that, but that's the easiest way. Any other questions?
So, cell, so the IgE molecule bonds to a cell.
It's already bound to the cell. It actually floats around bound to the cell. And then when it comes in contact, so let's draw it up here real quick, Shane. So we got your mast cell or your basophil. Right, it's going to have the IgE already attached. We'll call this a basophil. Right, so now what's happening is that it's actually it's got its two variable regions hanging out for it to find whatever it might be looking for. It binds what it's looking for, sends a signal, basophil releases histamine to go after it. Whatever it is that it bound to. So that's basically its trigger. Make sense? Any other questions? Seem a little bit more calm today than we were on Tuesday. All right, so clonal selection theory. This is the idea of how, kind of what we were just talking about. How is it that all these different B and T cells come about? What is it that they actually go through to actually become a mature T or B cell? And so this is the clonal selection theory. Remember when we use the word theory in science, that's about as close to truth as we can get, right? Law is the next step. But theory is pretty much, had most of its bases covered. There's really no other explanation. This is what most scientists believe. So able to recognize a limitless variety of antigens, right? Remember I told you that the number is, if it's 10 amino acids is all it takes, but there's 20 amino acids, it becomes 10 to the 20th power, so it's 10 with 20 zeros on it. So while it says limitless, well, technically it's 10 to the 20, but that's pretty high. that's a pretty ridiculous number. Each cell is specific for only a single epitope. Remember the epitope is those 10 amino acids. Diversity of antigen recognition is randomly generated. Right? This is what I was just saying. There's really no explanation to why the cell decided or why the hematopoietic stem cells make this T cell today, this B cell tomorrow. It just ends up being the way that that evolution designed the system. Lymphatic, circulating, residing, and secondary lymphoid organs wait to encounter the antigen with the correct epitope. We talked about this on Tuesday, is that most of the lymphocytes are hanging out in the secondary lymphoid organs, right? They're hanging out in the lymph nodes, spleen, places like that. The macrophages and dendritic cells then know that's where they need to go. So then they bring them back to that area to hopefully find the right B or T cell that has the correct ability to bind to that antigen and start the immune system response. Specificity of antigen receptors dictates epitope recognition. So the higher specificity, the faster it's going to recognize. So maybe it recognizes 8 of the 10 amino acids. That's not perfect, but it's a good start. And we're going to see this clonal selection theory then goes into the role of how those cells then become smarter. They become better at their ability to then bind all 10 of the amino acids that make up that epitope. But confirmation from another cell type is usually needed. That's the T-dependent ones, right? The majority of the antigens we're talking about in this chapter are T-dependent because we need that T helper cell to come in and help with the recognition. Well, that's why it's saying usually. Usually, the only ones that it doesn't are the T-independent, those ones that we've already been talking about ad nauseam. Upon activation, B cell then begins to multiply and then ultimately becomes a plasma cell. Yep.
So which one helps? With the B cells and T cells help the lymphocytes?
Those are both lymphocytes. So the T and B cells are lymphocytes. So that's like a class. All right. The B cells are the ones that are producing the antibody. T cells are the ones that are coming in and helping activate. All right. So here, this is a picture that's just showing you the idea of what's happening with the clonal selection theory. We have the hematopoietic stem cells up at the top. So going back to the very beginning where we were talking about the hematopoietic stem cells diversifying out into the different types of cells. Well, here, during development, it has developed 4 different cell types, B cells. Right? So they just got them listed up there, W, X, Y, and Z B cell. Each one of them has been able to recognize a different antigen. Well, these are what we call naive B cells. Naive B cells have not come in contact with their antigen yet. They've been produced. They have been made by the hematopoietic stem cell, but they haven't actually run into anything yet. Then they get activated. Well, in this case, the only one that got activated was B cell X. right? The second one there. It found its antigen, becomes activated. It needs a T helper cell, which is saying right up here in the note that it's not shown, but it's going to be shown in a different figure. But here, this wouldn't happen unless it was activated with the T helper cell's help. But what it is showing you is that B cell found its antigen, and now it starts to multiply, right? That 1 becomes 2, 2 becomes 4. They start producing, they start maturing into plasma cells, and then they start producing a ton of antibody, right? So that's going to be kind of the idea of the colonial selection theory. They're selecting the correct B cell based on the ability to bind to its antigen. These other 3, W, Y, and Z, they'll probably end up just dying off because they never ran into their antigen, and they'll go away and new ones will be created behind them. Just so happens in this case, B cell X was there. It was recognizing the antigen that was around, started producing a whole bunch of itself. It then goes into plasma cells. Plasma cells are then producing antibodies. But they all started as an immature B cell. All right, so here's the role. Here's those different classes or the different stages that they go through. Right, so I've used these words a lot, but now you got a slide here to show it to you. Immature. They lack fully developed antigen-specific receptors. So they have been created, but they don't have their receptor up on their surface yet. So in this case, these are just lymphocytes, so this could be B or T cells. They both go through the same role. So they first come out of the hematopoietic stem cell. They don't have the receptors yet, so they're immature. Naive lymphocytes have the receptors now, so they now have their receptor that only goes after that specific antigen. Right? Each receptor is going to be different. But now they have the receptors, but they haven't encountered that antigen yet. So they know what they're looking for, but they've never found it. Right? Activated lymphocytes, they have now bound to their antigen, received the co-stimulation help they needed, and then they're able to proliferate. They become activated. So they've found their antigen now. They got the signal that, hey, this is the right one we're supposed to go after. They start replicating, and then ultimately they become what's called effector. Effector are actually doing their job. So in this case, a B cell becomes an effector cell when it becomes a plasma cell.
When you say they receive co-stimulation, does that mean that, is that basically like the signal that they're evaluating?
Yeah. Yeah, because we'll also see that sometimes they grab, they also sometimes will grab the wrong thing, and then they don't get that stimulation help.
Okay.
Yeah. Did you have a question?
I'm sorry, you said that the effector lymphocytes are the ones actually actively
Are doing their job.
Okay.
Yeah. Well, and when I mean doing their job, they're clearing the infection. Right. Then once the job is done, so that's what it says here. Effector lymphocytes are descendants of activated lymphocytes. Either they become plasma cells. Plasma cells are descendants from B cells. So B cells ultimately become plasma cells. The T cells, they either become cytotoxic T cells or CD4 helper cells, right? So both of them started out as just a generic T cell. So that's what it's saying, they're descendants of. So there's no such thing as a cell. There's no such thing as a lymphocyte just jumping in and becoming a plasma cell. They have to go through this process, just like we mature from child, from infant to toddler to child, on up. These are the stages that the cells go through. Once the job is done, provided that the job was successful and they cleared the infection, the effector lymphocytes aren't needed anymore. We don't need that huge army of plasma cells and T cells anymore because that antigen is gone. Whatever that infection was, it's gone. So only a few of them are going to then get left behind as memory cells. And they're going to be there to remember that particular infection, so that way if we see it again, we won't necessarily get sick again. So as we're saying here, are long-lived descendants of activated lymphocytes responsible for rapid secondary response. Yes, ma'am?
The TC cells, you said those are cytotoxic.
Cytotoxic. So those are,
they make cytokines or what?
no, they all make cytokines, but the cytotoxic, so when we use the word cytotoxic, it means toxic to the cell. So they're the ones that are going and causing apoptosis. So think about the ones that have a virus inside them. The cytotoxic T cells go, recognize that cell is infected, and ask it to kill itself through apoptosis. So it's cytotoxic. It's causing that cell to die. Yeah. Make sense? Yes, Shane.
What is the main difference between activated and the effector?
Activated is that they have found their signal or they found their antigen and that they have proper confirmation. The next step is then they become an effector. So it's just saying when they become activated, it just says that it's just in a brief moment that they become activated. They're still not doing their job yet, but they now know what they found is what they're supposed to be finding. And now they know to go become, they differentiate into their effector cell and start doing their job. Yeah.
That was a good question.
Okay, cool. All right, so B cell activation by T-dependent antigens. Remember, these are the ones that require the T cell's help. Okay, so the B cell receptor binds to the antigen. So that's step one. B cell comes along. Remember, it's just going to have these are just the ones that we were just talking about before, not the naive ones that are essentially have their receptor, but they haven't run across anything yet. So we got our, of course, we got our B cell. That's why I keep a million of them in my pocket. Got our B cell. Got its receptor on it. Right, so BCR is B cell receptor. Right, so it's happy. It's a naive. It's never found its antigen yet, but it's there. The B cell receptor binds to its antigen. So now it has its antigen bound to it. Right. So the antigen is internalized. So at this point, now the B cell receptor goes inside, gets processed, and the B cell is then going to put the antigens out on an MHC class 2. Right. So now the antigen is going to be out here on MHC class 2. So the B cell's receptor's job is to discover the antigen. Once it discovers that antigen, it internalizes it, processes it through basically breaking it down through the lysosomes and all the other digestive enzymes that we talked about, breaking it up in all those little pieces and parts, and then it throws it up on the MHC class 2s. Right? So the MHC class 2s are only found on the antigen-presenting cells. We talked about them briefly on Tuesday. So remember, we got MHC class 1s. Those are found on all nucleated cells. So any cell that has a nucleus in your body has an MHC class 1. So it's kind of like your self-address, right? That's the ones that recognize. That's what we're looking for when we're recognizing self versus foreign. MHC class 2s are found only on antigen-presenting cells. And this can be B cells, dendritic, or macrophage. Okay, so those are the three major antigen-presenting cells. So that's why it's saying MHC class 2. Fragments of delivered MHC class 2 molecules presented on the B cell surface. So essentially it's internalized. The B cell receptor has found its antigen, internalizes it, and then puts them back up on an MHC class 2. Now it's an antigen-presenting cell. It's going around trying to find a T cell to ask, is what I got here in my MHC class 2, is this what I'm supposed to be going after, or is this something I shouldn't go after? So it goes and it finds its CD4, right? So remember also, with your CD4, these are the helper T cells. CD8s are the cytotoxic. Okay, so the CD4s are helping. And so the CD4 is going to go, and it has its T cell receptor on it. It's also a naive CD4 cell. It hasn't found its antigen yet. It's getting ready to when it binds to MHC class 2. Binds to MHC class 2, binds that fragment. T cell activates the B cell by co-stimulation. So if it gets this and it recognizes that antigen, it says, good job, B cell. It'll send a positive co-stimulation to to the B cell. That B cell then turns into an effector plasma cell and starts producing antibodies against that antigen. And so in this case, the CD4 T cell has recognized what the MHC class 2 has in it, sends a positive signal, tells the B cell good job. B cell then knows it's good to go ahead and mature further into an effector plasma cell, start producing antibody. If no helper cell recognizes a peptide, so it goes and it goes around looking at all the different T cells. It might be in the lymph node or it might be in the spleen. It's looking for a partner. If it doesn't find one, the B cell basically becomes what we call anergic, which just means unresponsive. It just kind of becomes a vegetable. It's sitting there with it's still got its antigens on this MHC class 2, but it didn't find a dance partner, so it just becomes sad and anergic and doesn't do anything. Yeah.
I'm not sure if I have this up. So is a B cell a plasma cell, or the B cell turns into a plasma cell and then differentiates itself?
Boom. So the B cell is a mature B cell, naive B cell, activated B cell, plasma cell, memory B cell.
Okay.
So the B cell becomes a plasma cell when it's doing its job. The only cell type that produces antibodies is a plasma cell.
And plasma cell meaning IgG,
IgA, IgM, all 5 classes of antibodies can only be produced by plasma cells. Does that make sense? Any other questions?
So when you're saying so after it becomes the plasma cell, then you said the memory B cell.
So it changed its name back to a B cell again. Why did it change its name to a plasma cell? We'd have to go talk to the guys and gals that decided that was going to be its name change.
I like it.
Right. It was a struggle for me to pick up on all these names too when I was first learning them, because it doesn't make any sense for it to change names from B cell to plasma cell and then back to a B cell again, but that's what it does.
This really is talking about B cell activation by T-dependent antigens. Is talking about the plasma cell. Once it's already turned into the plasma cell, then this starts to happen.
No, this happens. So it's all right. Let's go back up here.
Or this is just like in detail explaining that.
It's in detail explaining this. Yeah, so basically from this point here, the naive B cell is one that hasn't come in contact with the antigen yet. But here, when I drew it up here, it finally came in contact with the antigen. It becomes activated, but it still needs T cell help. The T cell help comes and activates it. If it finds what it found is good, then it gives it a thumbs up and says, go ahead and move over and become a plasma cell. And if it says no, or just ignores it, then that B cell never makes it past finding its antigen. It never becomes activated. It never becomes an effector. Yeah, so it just stays, basically becomes unresponsive. That make sense? Sure. Yes. Plasma cells are the only ones producing antibody, and that's good. We don't want them producing antibody until we're absolutely sure that what it's going after is what it's supposed to be going after.
So if it doesn't become the effector, does anything come in and kind of like?
Eventually, it'll die off. Like within a few days, that B cell that's holding on to that antigen that nobody liked, it'll eventually just die off, as all cells do. Most of our cells only have a short life period. Like our skin cells are only 7 to 14 days. Our immune system cells are only a couple weeks. It will. Apoptosis is the right way for them to die. Yeah, it'll be through apoptosis. But is there a process for that to happen? We're not getting into all that yet. We're not at all, actually. We're just going, it's hard enough already just to figure out how it all comes on. So, again, that's really all we're doing in this chapter is showing the on switch to the immune system, which hopefully you're appreciating is incredibly complicated, right? Now, hopefully, you can appreciate all the side effects of some of the drugs that you see out there. It's like, this may cause 52 other things. I've seen one today or yesterday. I said, if death is one of your side effects, see a doctor. I'm like, wait, what? I hit the rewind button on the DirecTV and go, is that what it said? But anyways, I digress. All right, so that's what we're talking about here. It's basically the on switch, right? The on switch to going after the T-dependent antigens. We need that T helper cell's help. T helper cell comes in. If the T helper cell recognizes the MHC class 2, because another thing we need to remind ourselves, just to try to keep putting all this together. B cell has already taken care of its receptor. MHC class 1s are recognized by CD8 T cells, CD4 T cells. So the students that missed that on the, I think those are gimme questions, those two, but CD8 bind to what? MHC class 1, CD4 is bind to MHC class 2. I think those are two gimme questions, but people miss them every semester, so try to get that stuff in your head. That'll help you also understand it. The CD4s are the ones that go after the antigen-presenting cells, or the helper cells. That's why they go after the MHC class 2s. All right, the last thing, results in tolerance to the antigen. So that's a good thing. That means the immune system has recognized that even though that B cell recognized that antigen and went ahead and pulled it in and put it out in MHC class 2, even though the immune system decided, the T helper cells decided, nah, we're not giving any extra help to that B cell. The B cell is a moron. You got the wrong thing. Then it ends up being tolerance. So we then can tolerate that antigen being in our body. Our immune system ignores it. It can be. Good question. She just asked, can that antigen still be harmful, though? Yeah, can be. So then we have to go in, and that becomes chronic diseases and things like this. It becomes problematic occasionally. Very good. So here, just a cartoon, hopefully explaining it just as well as I've been trying to. One, B cell receptor, boom, finds its antigen, is showing it as a Rubik's Cube. Why? Because it's showing that each of those squares is a different epitope. Right? The epitope is just part of an antigen. So here it's showing that Rubik's square. It internalized it, right? Because it recognized the antigen, so it brought it in, breaks it up into all the different pieces. Now it's out there in those MHC class 2s. All the different epitopes from that one single antigen are all out there on this different MHC class 2s. If it's something that's dangerous, again, as she asked, could it still be, even though it was ignored? Sure, but let's just go from a generic standpoint that it is something dangerous. The MHC class 2s are recognizing it, are out there, and then the T cell receptors recognizing that inside the MHC class 2, right? That's the only way it does it. And then it's sending a positive signal, in this case, cytokines, right? Cytokines are the voice. So it's sending the voice, it's saying, hey, good job. Sends a cytokine out there, tells that B cell to go ahead and further mature into a plasma cell. T cell's done at that point in terms of activating the B cell. Over here, if it's harmless, T cell ignores it. Right? Now we get tolerance to that particular antigen. All right, so we're moving around to characteristics of a primary response. So what is it? Again, these are just common things that everybody goes through, the immune system goes through when we're going through a primary response. 10 to 14 days is about what it takes for antibodies to accumulate in large numbers. So this is why you may be sick for a few days, because the first 3 or 4 days that you're sick, you don't know you're sick usually. And then you start getting those symptoms where malaise, which is just kind of not feeling all that great. You wake up and go, I don't feel that great. And then within another day or two, if it's actually progressing, then you actually get full-blown sickness symptoms. But usually by that point, you've been sick for a good week or better. It takes about 10 to 14 days for any substantial antibody response to occur. So that's why you get a cold, you may be sick for 3-4 days. By that time, your antibodies have finally started accumulating. You're right around that day 10 mark for a common cold. And then ultimately you get your antibodies, they start doing their job, get rid of that virus, you start feeling better, your immune system has taken care of it. Person may feel sick while primary response is developing, right? Just like I said, some activated B cells continue dividing, others differentiate to form antibody-secreting plasma cells. That's just going back to the idea of what we were seeing there with W, X, Y, Z B cells. That, if you go back to that picture, right here. Remember here showing that they start dividing to make their numbers bigger? That's happening too at the same time that some of them, because after we start getting an army of those B cells, remember it's just that one B cell that was able to recognize the antigen, starts making copies of itself. That's why it's called a clonal selection. Which clone are we picking? It has to be activated, or it has to be recognized and then activated. We got to keep making more and more of those B cells to ultimately make more and more plasma cells, But we go ahead and allow those ones that have already been made to go ahead and differentiate into plasma cells so they can start producing antibody. But this is still happening too. We're still activating more. We're still making more and more of them while we're also allowing them to move forward to plasma cells because we got such high numbers at that point. So that's what that is referring to here. Some of them are going to continue to divide while others go on to become plasma cells. Makes sense because we got so many at that point. And so look here, IgM. Those are those two little mounds you see right here. Primary response on your left, secondary response on your right. First time, second time seeing a particular antigen. Days versus months. So in this case, primary response, the antigen comes in right away, right here where the X and Y axis comes together. You get an IgM response, but IgM never goes very high. Remember, it can't leave the bloodstream. It's that big pentamer thing. It's out there looking for those T-independent antigens. So it'll come up in a little bit higher numbers. We'll see a little bit of a surge in them, but not a lot. What we do see, though, is we see that little peak of IgG popping up, right? I told you IgG is the most important antibody for going after a response. And so IgG pops up, starts weaning, right? Because this is essentially this weaning that we're seeing happening here. That's essentially after the response has been dealt with, after the pathogen has been dealt with. We don't need those high numbers anymore, so it starts going down. But look what happens the second time you see that antigen, which is astronomically higher than the primary response. That's because those memory cells are already there. They already know how to deal with it. They just automatically get to differentiate straight back into plasma cells. They don't have to go through the immature, naive process per se. Memory cells, they have the B cell receptors out there. They recognize the antigen, they're a memory cell, they automatically just start going to plasma cell. So they can have that secondary response happen much quicker.
I have a question about this side, the last slide, the MHC. Those aren't considered like a receptor?
They are receptors.
Oh, that's not that. They look like the little nodes.
Yeah, they're receptors.
You call them like molecules instead of receptors.
Well, molecule is a generic term for anything. So yeah, they.
I thought they were like,
Yeah. It's a lot of vocab for sure, and understanding when certain words basically mean the same thing. Yeah.
When the CD4 cells give their thumbs up, whatever, do they stay attached, or do they just continue floating?
They keep floating around and go activate more B cells if there's more floating around. So yeah, they just come, they make that initial handshake with the B cell, tell them do their bad job, and then they move on.
So the number of CD4 cells, CD8 cells that you have is going to be lower.
No, not necessarily.
You have a lot of them.
Yeah, and you got quite a few because each T cell can only recognize a certain B cell also. So they can only recognize a certain antigen. Just like the B cell receptor can only recognize a certain antigen, the T cell receptor can only recognize that antigen too.
Okay.
And so yeah, you gotta have just equal numbers pretty much.
So T cells are always floating around?
Yeah, same thing with B cells. They're just floating around constantly. They're constant. Yeah, usually they're hanging around in the lymph nodes and the spleen because you want them all in the same location. So that way, when the macrophage or dendritic cell comes in with that antigen, it knows where to go and it has a maximum chance of running into.
So it already knows it doesn't
Yeah, it's pretty wild when you think about all these things truly happening in your body, right? It's just pretty wild to think about it all.
I have a question.
Yeah.
When lymph nodes have to be removed, like cancer patients and whatnot, what causes lymphedema? Because they said that's the fluid builds up because of the lack of having a lymph node. So how
it
does
sort of reminds me, I'm always thinking of like analogies, to be able to make sense. Say you live outside of New Orleans and they suddenly close I-10 West. Makes it really good. It's going to cause all the secondary roadways to become clogged and everything's going to be just stressed out, right? Everybody's going to be in a big traffic jam, right? You start removing lymph nodes, same thing's happening. You have less roadway essentially for the T and B cells to make their way through. And so now you're going to end up getting clogs. And so, yeah, there is a lot of like lymphatic massage therapy, things like this, helps massage through the lymph because the lymph is very syrupy, and so it can start clogging up, especially if you have a massive infection like cancer or sometimes they even have bacterial and viral infections. You have so many B and T cells and antigen-presenting cells all trying to get into that lymph node at the same time. It ends up causing a clog, or where the lymph node then starts to become atrophied and starts to die. And so then you have secondary problems. Yeah, does that make sense?
So the capillary beds then just can't handle.
They just can't handle all that happens.
So everything stays in the capillary beds and tries.
Or it gets stuck in the lymph node, or it gets stuck in the lymph nodes that are still present, and then it ends up just starts swelling that lymph node up. So think like tonsils, especially for children that end up having to get their tonsils removed. The tonsil is a lymph node, but it is a version of a lymph node. Why does it swell up? Because children are getting infections all the time, right? Because it's the first time they're having a lot of primary responses happening in that age group. And so, the age group that you end up having, kids need to get their tonsils removed, adenoids removed. The tonsil has essentially flared up because it's just so full of antigen-presenting cells, B T cells, all this stuff that ends up just becoming too full, and then it starts cutting off the airway, cutting off the ability to swallow properly. And then the best thing to do with current technology is to remove it, which, like I said, I really don't like that idea, but when you're dealing with life.
That's what I was going to ask, by removing it, doesn't that happen when you get the fluid build up in other places?
Yeah, that's what
There's no way
I don't
just
like it, but
drain
I've
or
never had.
something like that. They can't just.
The one or two doctors I've had a conversation with that actually do the surgery, they're like, we get paid double. We take the adenoids out at the same time we take the tonsils out, and it's just as easy to take them out. But their argument is not only is it a payday for them, but also that it helps avoid any secondary surgeries if the adenoids become clogged next.
Or you just keep removing stuff and then it's going to build up somewhere else.
Now you're on the same page. Again, that's my belief. I mean, I'm sure there's somebody out there who would argue just as ferociously next to me as telling me I'm wrong, but that's my belief. Yeah, Shane.
In the chapter, it said that CD4 had to do with HIV.
Mm-hmm.
Is that because, real quick, is that because people who acquire AIDS, is it because that their body is not producing enough CD4 to produce antibodies to kick the disease out?
You're close. So what happens, and this is good because I'll bring it up multiple times in the semester because it is one of the best stories for CD4 T cells. CD4 is the receptor that HIV is looking for. So they actually, the only cells, one of the only two cells that HIV actually attacks in a patient is CD4 T cells. And so it goes and just starts shredding your immune system. I don't remember I'm recording this. Shredding the immune system apart. So now you don't have those CD4s that come in and help anymore. Because over time, especially in the beginning when HIV first came on the screen in the 80s, people died within like 6 months to a year sometimes. Well, if I basically wipe all your CD4 T cells out of your body, you don't die of AIDS. You die of any secondary infection you get because your body doesn't have the ability to fight it off. Yeah, like pneumonia is a good one, but even one of the weirder ones is like Cryptococcus pneumoniae, or Cryptococcus neoformans, which is actually a fungus found in pigeon dung. So walking around in a city like ours that has lots of pigeons, we're breathing in it all the time. Our body's able to take care of it. It's a non-issue. Somebody that doesn't have CD4 T cells, like an an advanced HIV patient, they have no ability to fight that off. And so that's because HIV's main cell target is CD4 T cells.
The reason I was asking is because I had a friend of mine passed away like a year ago.
Mm-hmm.
He survived 38 years
Mm-hmm.
of HIV, but towards like the last 6 to 7 months, no matter what medicine he was on, would help. And within like 6 months, he went from like 275 pounds to like 130 in like 6 months. So I was just wondering,
Think about it as a balance. So we're trying to balance your CD4s with the virus. And so in the beginning, the virus just goes and wipes all the CD4s out, virus wins. The new medicines we have try to keep that balance in play by protecting,
There's
covering.
no, like, synthetic form of CD4?
No, what, the medicines? Medicines are actually one of the. We're getting a little tangent, but I don't mind because it actually makes sense for what we're talking about. MHC, so like one of the medicines that we use is an entry prevention medicine. It blocks the CD4, so now the virus can't see it. It blinds the virus to CD4. That's one of the drugs we use. The other drug we use prevents the HIV from actually replicating itself. And so even though the virus might get into the cell, it can't replicate. So again, we're trying to keep that balance where the CD4s can constantly still maintain, right? Even though they're getting the crap kicked out of them constantly. What happened with your friend probably is age. As you age, your immune system starts weaning too. How old was he? 63? Yeah. So his deal was that as you start aging, your immune system isn't as good at making more T cells and B cells. So hematopoietic stem cells start slowing down. And so now there's not as many coming behind to fill the void of the ones the virus is taking over. And so quickly, as you pointed out, 6 months or so, the virus is just winning, right? And once it starts winning, we're done. Conversation's over at that point, for that, unfortunately.
So if you were to eliminate all somebody's C4 cells, the virus couldn't hang out in the body, correct? Or it could hang out in the body and could host off the body, it just wouldn't.
Yeah, but you wouldn't live very long.
How do you get it? Because you need your C4 cells
to fight off infections.
Right.
You need any T-dependent infection needs the CD4 T cells to activate the B cells.
So it's not like we can just go in there and kill somebody's C4 cells.
So if I got rid of this, the B cell would never get its secondary signal that it needs to produce antibodies, right? So yeah, in theory, sure, if we got rid of all the CD4 cells, and the HIV couldn't really replicate, but then the person wouldn't be able. Yeah, you need them. You can't go without them, right? It's like, hey, I got this brand new Ferrari outside, but it doesn't have an engine, right? There's nothing you can do with it, right? So it doesn't really help anything. Sorry, my analogy sucks. All right. Anybody else got any other questions? Good question. I appreciate you trying, because that's critical thinking. That's actually trying to put together all the pieces and parts and make it make sense, versus just sitting and reading all these things.
I think this is one of the only times I've actually put all the steps together and just studied. Any other time it's like,
it just takes time. It's usually about week 4 or so. You all start picking up, This isn't new to me. It's I see this every semester, and see students struggle, and all of a sudden the light bulb starts clicking in and start making sense of things. We got about 10 more minutes, so we'll keep moving forward. All right. So we talked about primary versus secondary. We get that much faster response because memory cells are there now, so it's much easier for them to get through. The next slide there is talking about some B cells differentiated to form plasma, right? We've already talked about that ad nauseum today. So we know that the B cells then move on after they become activated, become plasma effector cells. The antibodies are then just released into circulation. They're activated, continues proliferating and differentiating in the presence of the antigen. So as long as the antigen is still in your body, those B cells just keep making more and more of themselves. More and more of them get turned over to plasma cells, and they just keep going after that antigen until that antigen is gone.
So that process basically, grabbing the T helper cells keeps going.
Just keeps going until the infection's cleared, which is a good thing. And so here's the characteristics of a primary response. This is what we call affinity maturation. So maturation is just maturing, right? So affinity is, how affectionate you are, right? In terms of how affectionate it is for a particular antigen. So like this is the idea of, remember the epitopes usually are about 10 amino acids long. And so let's say the B cell recognized only 7 of the amino acids. It's pretty good. That's a pretty good match. But it's still not 100%, right? And so what's going to happen is through a process that evolution has given us, natural selection has given us, it is going to go through this process where it's going to, every time it differentiates, every time it proliferates, I should say, every time a B cell goes through replication, it's going to quirk. It's going to mess with the genetics just a little bit of the binding site. And so sometimes it makes one that's not as good. So the very first one is pretty good. They gave 2 positives. I mean, it's just trying to demonstrate something here. So 2 positives. It's a pretty good match. But yeah, when it went through its first round of proliferation, it made one that was not as good. That one just kind of, it just, it dies off through apoptosis, whatever. It's not used. This one here, it's a better match. It goes through a second round of proliferation. Well, now it's made even a stronger affinity to that antigen. It's still the same main B cell. It's still recognizing that same antigen, but now it's becoming even more affectionate to it. It's able to grab it even stronger. It's a better response. And so now that one that originally was good is not as good anymore. So now this one becomes the one that gets a signal to keep proliferating. And so over time, the point being, over time, the immune system becomes even better at recognizing particular antigens. Because every time it goes through proliferation, it's twerking or tweaking the genetics just a little bit to see if it makes a better match, which is what it's trying to do. Sometimes it's a worse match, and that one's just ignored. But eventually, at the end, we end up having B cells with the highest affinity, most likely to continue proliferating. So they're the ones that we're picking, right? We don't want to pick the other ones. We want the ones that are the best to be the ones that we continue moving forward with. So proliferating B cells help improve the immune response, right? So the immune response gets better and better over time. And then eventually we're going to have class switching happening. So remember the IgA, D, E, G. Remember the IgMs are part of the primary response. So if we were to draw it up here, we got our first and second response, the primary. First one here is always going to be IgM. Right? IgM are the first ones made, but remember they can't leave the bloodstream. They really are limited because they're a big pentamer. They're mainly after looking at T independence, but that's what the plasma cells are normally secreting. The helper cells then come in and activate the B cell. They become a plasma cell that secretes the other antibody classes. So once the T cell comes involved, we'll call it the CD4, becomes involved, then the IgM switches over, then the plasma cells start producing IgG, IgA, IgE, and even probably D. Right, so the CD4 tells the B cell which one of those four to start producing. So it switches from IgM to those other four. Secondary response.
Start off with
IgM is always part of the primary response. It's always circulating, even when you're healthy. When you're healthy and you haven't been sick for a while, because we do have that half-life of the IgGs is pretty high. We've seen that weaning of like the child. The child weans after about 6 months.
So the CD4 tells the IgM to differentiate into one of the other.
Tells the plasma cell. Plasma cells what's making the antibodies, right? So plasma cells that are just circulating normally in your body are producing IgM in case it comes across a T-independent antigen, right? So they're always out there. But now that a B cell comes in and says, hey, we actually got a serious problem over here, and then the CD4 cell will send a signal to tell it to class switch. So that's all that class switching is switching from producing IgM to one of the other ones that are more important. So if it's a mucosal problem, it's going to produce IgAs. If it's a allergen problem, it's going to be IgEs. And if it's pretty much anything else, it's IgG response.
So CD4 tells the plasma cell which one to.
Mm-hmm. Exactly. But yeah, but questions that I would ask you. So again, I know there's a lot of vocab in here. Focus on the question, right? What would the question be? Class switching. What is class switching? Class switching is changing what the plasma cell is making from IgMs to one of the other four responses based on what the antigen is, right? Don't get lost in the weeds trying to understand it all. Because I didn't even really truly get into the weeds of immunology until grad school, right? Yeah, so a lot of this, can really get complicated pretty quickly. Focus on the surface details. All right, so B cells in lymph node produce the switch IgG, right? Because lymph node is going to be pretty much anything and everything you got going on in your body. But if it's in the mucosal-associated lymphoid tissue, it's going to switch to IgA, right? Because it's mucosal. Yeah, so it just depends on where it's at.
So, sorry, a good way for you to sum that up. You said changing what the plasma cell is making based on what it needs.
Mm-hmm. Yeah, perfect. And instead of switching over, I assume what the next slide is, so I'm not going to switch over to anything more complicated with only 5 minutes left. So what we'll do is we'll stop there. Obviously, we're behind schedule, but that's cool because, like I said, when I was telling you when we had to push the exam one and make sure we got it taken care of, I knew this stuff was coming up, so I didn't want to dedicate any extra days to the easier stuff. But I knew that immunology was coming up, so if we fall behind a little bit, that's fine. I'll sort it out and let you know any changes that we make. But right now, yeah, we're going to leave it there for now. But I'm just saying, in case we fall behind.